my YouTube channel. I'm Dr. Poonam Agarwal, and in this session, I'll be talking about the key aspects of molecular biology and genetics, which is going to be very important for your various upcoming exams. So let's start the discussion. And first of all, I would like to talk regarding the structural aspect of the DNA, where the Watson-Crick proposed a model for the DNA in 1953. So this is important topic. A lot of questions are framed against it. The key aspects of this Watson-Crick model was that DNA is a double strand. DNA is double stranded and both the strands are anti-parallel. Both the strands are arranged in an anti-parallel fashion. They are not parallel. They are anti-parallel. And it's a coiled structure. What they said, it's a coiled structure and you have major and minor grooves. It's a kind of coiled structure and one complete coil, that is 360-degree turn of double-stranded DNA, covers the height of 3.4 nanometer. You can see this is the height, which is covered completely in a major groove, and this is going to be 3.4 nanometer. In other words, it is in other unit, it is going to be 34 angstrom. And they said that in one major groove, one complete turn of double-stranded DNA, there are 10 nucleotide pairs which are going to make the steps of the ladder. So in one major groove, which is covering the height of 3.4 nanometer, you have got 10 nucleotide base pair. So if you talk about the distance of one base pair in this double-stranded DNA, it comes to 0.34 nanometer, which is going to be 3.4 angstrom, right? So these numbers you should remember. A lot of questions are framed, and if you get a short note in your prof examination, you have to write all these things. So they said double-stranded DNA are anti-parallel, and backbone is phosphodiester bond, right? So it's a phosphodiester bond which is making the backbone of the DNA, and the bases are projecting in the space in between these two phosphodiester backbone. The bases are projecting in the groove in between, and they said that guanine is always going to pair with cytosine and adenine is always going to pair with thymine. So, in other words, purine is always going to pair with pyrimidine bases. So, depending on the number of purine bases, you have equivalent amount of pyrimidine bases, pyrimidine nucleotides, or you can call it as pyrimidine bases in the double-stranded DNA, because A is always going to pair with T, T is always going to pair with G. So adenine and guanine, they are purine bases, right? Purine nucleotides. Adenine and guanine, they are purine bases. They are present in the purine nucleotides. Thymine, cytosine, they are going to be pyrimidine bases and pyrimidine nucleotides composition. So the purine nucleotide bases are always, connected with the pyrimidine nucleotide bases. So that is why you can see even in this picture, there was a time in previous year they asked one question that what is these colored lines representing? You have to say they are bases. And what is the link which is linking the guanine to cytosine or adenine to thymine? You have to say it is hydrogen bond. So hydrogen bond. You have two hydrogen bonds between A and T, and you have three hydrogen bonds between C and G. So the portion of the DNA which is having rich amount of G and C, of course, is annealed tightly, so it will be needing higher temperature for separation. And compared to that segment of the DNA which is having rich amount of AT, because they have weaker hydrogen bonds. AT has weaker hydrogen bonds. Lesser temperature will be required to separate both the strands. So these points you must remember. And this picture is talking to you about the basic structural aspect of a polynucleotide, which is DNA or RNA. And in this picture, you see this is the structure of DNA. So polydeoxynucleotides, right? So it's a deoxynucleotide, polydeoxynucleotide. Remember, deoxynucleotides are added together through phosphodiester linkage. So if you talk about a nucleotide composition, you have three components in a nucleotide. You have got a base, you have got a pentose, and the third component is going to be the base may be purine base or it may be pyrimidine. Pentose may be ribose or it may be deoxyribose, right? So the difference between ribose and deoxyribose, oxygen is absent. Second carbon hydroxyl oxygen is absent, minus second hydroxyl. So in deoxyribose, you do not have second hydroxyl. And in ribose, you have second hydroxyl. That is the only difference. And if the pentose is deoxyribose, the nucleotide will be deoxyribonucleotide. And if the pentose is ribose, the nucleotide is going to be a ribonucleotide, which will be the composition of RNA. Phosphate number will be 1, 2, or 3. If it is 1, you call it as a nucleoside monophosphate. If it is 2, you call it as a nucleoside diphosphate. If it is 3, you call it as a nucleoside triphosphate. But everything is a nucleotide. So when you say nucleotide, you just do not know whether you're talking about 1 phosphate or 2 phosphate or 3 phosphate. If you want to specify the number of phosphate in a nucleotide, you have to spell it as nucleoside. Nucleoside is nothing but it's the base and pentose together. In that, if 1 phosphate is there, it's called monophosphate. If 2 phosphate is there, diphosphate. 3 is there, triphosphate. ATP is triphosphate. So whether it is monophosphate, diphosphate, or triphosphate, it is always a nucleotide, right? So that difference you must understand between a terminology of nucleotide and nucleoside. And I picture, it may project in your exam as an MCQ. They can ask you, what is this structure? You can see this is a polynucleotide. Now let's see how it is a polynucleotide. If you focus on this portion of this picture, this is one nucleotide. And if you focus on this portion of this picture, this is going to be another nucleotide. Do you understand why I said it's a nucleotide? Because if you see, this is a nucleotide, this is the base, right? And this is the pentose, which is deoxyribose in this case, and this is single phosphate. So it's a nucleoside monophosphate, deoxynucleotide, right? So similar type of, the other bracket also is showing you a deoxynucleotide. You see, this is the base, this is the pentose, which is deoxyribose again, because again oxygen is not there. Phosphate, right? So this is how a lot of nucleotides, deoxyribonucleotides, you can see, and you can see that they are connected to each other. Now the question is how they are connected. You can see this is the bond. Bond formed between third hydroxyl of existing or initial nucleotide and fifth phosphate of next nucleotide. This is the third hydroxyl and fifth phosphate. This is fifth phosphate, right? This is third hydroxyl. It's the interaction between these two which is going to create a esterification, and this bond which is produced is actually going to link the nucleotides together. And this bond is called phosphodiester bond. It's called phosphodiester bond. Phosphodiester bond. To be more specifically, if you want to say polarity wise, what is the correct name for the phosphodiester bond which links the nucleotides together to make you polynucleotide, you must know it is 3 prime 5 prime phosphodiester bond, because it's always going to be third hydroxyl and fifth phosphate which is going to make you phosphodiester bond. So these are certain little critical points regarding this kind of structure. If projected to you, you must identify where is the base, where is the pentose, and what sort of pentose is there. In this picture, it is certainly deoxyribose, right? So it's certainly deoxyribose. And why? Because the pentose is not having oxygen at the second carbon position, right? So this is deoxyribose, right? Now let's proceed further, talking about nucleosome. What is nucleosome? You may write nucleosome is a primary level of DNA condensation. Primary level of DNA condensation. So DNA, you imagine it's a long thread, double-stranded thread, but when you see in the nucleus, it is present in the chromosome form. So what is the difference between DNA to chromosome? Chromosome is nothing but it's a condensed DNA. Many-fold the DNA is getting condensed to make you actually the chromosome, small space within the nucleus. So whenever we are talking about the condensation of DNA, the primary level of condensation, where is first the DNA is going to wrap around the histone octamer, making you the nucleosome. This is one nucleosome. Do you see this? This is one nucleosome. Nucleosome is an important target for MCQ pattern of examination as well as for professional examination, where they draft long answer, short answer questions. So whenever you're talking about nucleosome, you have two important basic components in it. One is the histone octamer. Histone octamer. Another is double-stranded DNA wrapping around it. And they say histone octamer is a histone protein which is having 8 subunits. Now they ask you, what is the composition of histone octamer? Spell the components of its composition of histone octamer. You have to say it is 2A and H2B, which makes the dimer, and two such dimer is present in this. I repeat, 2A and 2B, they make a dimer, and two such dimer is there in this. So this is equivalent of 4 subunits, isn't it? Then similarly, H3 protein subunit is there, and that too 2 in number. So this is again going to make you 2 subunits here, right? And H4 is also going to be 2 in number, so it's going to make you 2 subunits again here. So it's going to be total 8 subunits. Going to be total 8 subunits. That is why it is octamer. You do not have H1, no H1 in histone octamer. I'll tell you where this H1 is lying. Shortly I'll tell you. But in histone octamer, you do not have H1. That's a very frequent MCQ to you. Now this is how you have histone octamer. In this picture also you can see all these names which I have just told you. 2A, 2B dimer and 2 such dimer, 3, 2 units, 4, 2 units making you histone octamer. Now can you see the spring-like structure which is wrapping around this histone octamer? This is nothing but it's double-stranded. And how much wrapping? Whether it is a complete 1 turn, complete 2 turn or incomplete 2 turn. So you must know that double-stranded DNA is wrapping 1.75 times around the histone, or 1.75 times, which is going to have 146 base pairs to at times 149 base pairs. Portion of double-stranded DNA only is going to wrap around the histone octamer. That is one complete turn and second incomplete turn. After that, the DNA proceeds ahead. Right? You can see in this picture that after wrapping the histone octamer, this spring-like structure is proceeding ahead to wrap another histone octamer. We can see two nucleosomes in this picture connected to a linker DNA, right? So you must know all these nomenclatures. This linker DNA, the size of linker DNA is variable. They say that number of nucleotides in this linker DNA may vary from 30 to 90 base pairs. It's not the length which is connecting two nucleosomes together. That number varies, 30 to 90 base pair, like the base pair will be there in the linker DNA. So this is how, this is primary level of condensation of the double-stranded DNA, and this structure is called polynucleosome. When you have multiple nucleosomes connected through linker DNAs, right, you call it as polynucleosome. You call it as polynucleosome. And then in this polynucleosome, you find the structure of polynucleosome very beautiful. It looks like a string, pearl on string appearance. It looks like pearls on string appearance. It looks like that, like pearls on string appearance. And if you talk this kind of rapid double-stranded DNA around the nucleosome, if you see the height of this nucleosome, it's going to be 11 nanometer to be exact. At times it see that the width which is covered in this fashion, or height which is covered in this nucleosome along with the DNA wrapping, is going to be 11 nanometer, but at times they write it as 10 nanometer. You remember it as 11 nanometer filament. It is also known as 11 nanometer filament. Okay, now coming to the next aspect of this elementary genetics, where I'll be talking about the heterochromatin and euchromatin portion of the chromosome. As you visualize in this picture, you have a chromosome. You can see short arm and long arm. You can see centromere, telomere. You can see the condensation, strong condensation. It's called heterochromatin. Loose condensation. You can see the loose condensation. It's called euchromatin. Euchromatin is less condensed. Heterochromatin is more condensed. More condensed. Rich in AP sequence. Euchromatin is rich in GC sequence. What is most important here, you must know that heterochromatin is having the condensed DNA. So the genes which are going to be there in the heterochromatin area are going to be silent. They will not be easily expressed. But in euchromatin, genes are expressing. You can remember E for E. Euchromatin expressed. And it's very logical to understand that in euchromatin area, there is a loose condensation of DNA. So the gene is getting easy approachability by various proteins and transcription factors and enzymes which are going to transcribe the gene, right? So, but in heterochromatin, that is basically going to be very difficult because that area of DNA is there in the dense condensation. The transcription factors and various enzymes are having no easy approachability. So that is the reason in heterochromatin genes are suppressed and in euchromatin genes are easily expressed, right? So these things you must know. And coming to next aspect, this picture is beautiful picture, but it's conveying the same information which I have just told you, that enzymes like RNA polymerase and all, they are having easy approachability in the euchromatin area and they express the gene easily. But in dense area heterochromatin, they do not reach easily. But what is Targar's role? Very simple. That you must know. Sargas was a scientist who said that A is always going to pair with T, C is always going to pair with G. And as I already told you, adenine and guanine, they are pure bases. And you can see they are always going to pair with pyrimidine bases. Thymine and cytosine, they are pyrimidine bases. So Sargas said that adenine is always pairing with pyrimidine. So in any double-stranded DNA, when bases are pairing, they will always pair in this fashion. So in a fragment of double-stranded DNA, the number of purine bases will be equal to number of pyrimidine. This kind of simple formula he proposed. Or in other words, you can see that in a double-stranded DNA fragment, the number of purine nucleotides or purine bases divided by number of pyrimidine bases is going to be 1. They are going to be equal. And this rule is applicable only for double-stranded DNA minded. It is not for single-stranded DNA. It is not for RNA. It is only applicable for double-stranded DNA because double-stranded DNA only we are talking about the base pairing. And he said that A will pair with T, C will pair with G, purine will pair with pyrimidine, pyrimidine will pair with purine. So that is what is the Targaf's rule. Why I want to tell you this? Because Targaf's rule can be it's paired in multiple fashion. You can say A pair with T, C pair with G, or the number of purine equal to number of pyrimidine, or you can say the ratio of purine bases to pyrimidine bases is equivalent to 1. Right? So all are correct regarding Targaf's rule. Okay, now coming to this next interesting topic, that's called denaturation of the DNA. What is denaturation? What is the other name for this? It's called melting of DNA. When you separate both the strands of the DNA, it's called melting. Generally, two strands of the DNA are approximated to each other, and the bond which is forcing them to get approximated is the hydrogen bond. So when you break the hydrogen bond, the DNA goes apart. And this is called denaturation or melting of DNA. When you use heat over a range of temperature, the DNA separates. Midpoint of that temperature is called melting temperature. If anybody asks you what is melting temperature, you see the midpoint of the temperature range over which the separation takes place. Melting temperature. So you can use heat to melt the DNA. You can use heat. You can even use alkali, even weak alkali, half normal sodium hydroxide, which is basically used in blotting technique, if you remember, that the DNA, that can denature the DNA. Heat, of course, we are using in PCR to separate the DNA. So heat also can separate the DNA. We call it as a heat denaturation step of the PCR, right? So that you must know. So increasing the heat, using the alkali, and one point you must know, decreasing the salt concentration also separate the DNA. So what does it mean? It means that if you increase the salt concentration, the annealing will be So you'll be needing higher temperature, isn't it? So using heat you can separate, using weak alkali you can separate, but decreasing the salt you can separate. So when you increase the salt, separation becomes difficult. So according to a calculation, when you increase salt concentration 10 times, the melting temperature increases by 16.6 degrees Celsius. So you must know the difference between the role of alkali and the role of salt as far as the DNA annealing is concerned. Alkali denatures, but salt strengthens it. So when you increase the salt concentration, it becomes strongly annealed. So that time you need higher melting temperature. Right. So this was about denaturation, and now coming to human mitochondrial DNA. You see, it's such a beautiful structure, double-stranded, circular, closed. Nuclear DNA is open-ended double strand, right? But mitochondrial DNA is a circular double strand. And in a zygote, the mitochondria comes from ovum. So whenever we are talking about mitochondrial DNA, it is donated by ovum only, which is, the maternal inheritance. Now, there are certain key points regarding human mitochondrial DNA which you must know. In a cell, you have 10 to 10 copies of mitochondrial DNA, and don't they are enough. They are very, 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 very important DNA which are having their whole system of making messenger RNA, transfer RNA, ribosomal RNA. The whole process of replication takes place here, in the mitochondrial DNA. Transcription takes place. Even the protein synthesis takes place in the mitochondria. So it's a whole organism itself. It's having its own DNA, own set of various RNAs which are important for protein synthesis. But one point you must know regarding human mitochondrial DNA replication is that it is without proofreading. So no proofreading during replication of human mitochondrial DNA, hence no proofreading, right? No proofreading. We have a strict proofreading while a nuclear DNA is getting replicated. But when mitochondrial DNA is getting replicated, there is no proofreading. So chances of mutation is very, very high in the mitochondrial DNA, right? And this human mitochondrial DNA contains 16,565 base pairs. This number also you must remember. 16,569 base pairs are there in the one double-stranded mitochondrial DNA. They produce a lot of proteins, but what is very important that they produce number of important proteins for the functioning of electron transport chain. According to a calculation, 13 proteins of electron transport chain are actually synthesized in the mitochondria by mitochondrial DNA, and then they are getting imported to electron transport chain. Rest of the electron transport chain proteins are synthesized de novo, from the nucleus, but 13 proteins are synthesized de novo in the mitochondrial DNA itself, and remaining proteins are synthesized and imported from nuclear DNA. One good number of proteins which is getting generated by mitochondrial DNA in the electron transport that you must know. And the same thing I have shown here, 1% of total cellular DNA is mitochondrial DNA, 2 to 10 copies of small circular DNA, maternal inheritance, 13 protein out of more than 80 proteins of electron transport chain is synthesized from the mitochondria, which is going to be approximately less than 20%. Now, what is central dogma of genetics? The DNA to RNA and RNA to protein is the central to this flow of information from DNA to messenger RNA, from messenger RNA to the protein via transcription and translation is constituting your central dogma of genetics. Replication, DNA replication is not the part of central dogma of genetics. It is only transcription and translation which is going to be part of central dogma of genetics. And you find amplification at the level of transcription as well as translation. Why? Because one gene is able to produce you multiple number of same messenger RNA, and one messenger RNA is capable of producing you multiple numbers of same proteins, So there is amplification in the central dogma. When we are talking about replication of DNA, there is no amplification. One DNA Parent DNA makes only one daughter cell, right? One parent strand, when they undergo replication, set of DNA is actually produced, but each will have only one daughter strand. So one will make two, two will make four, four will make eight. As such, there will not be any number of DNAs produced during replication, right? So there is no amplification in replication of DNA. And of course, the replication doesn't constitute the part of central dogma of genetics. When we talk about genetic code, it is three nucleotides which are going to constitute one genetic code. Now, there are certain characteristics of genetic code which you must know. They are made up of three nucleotides, yes, and they are being read in a non-overlapping fashion. Suppose if you're talking about one messenger RNA, you have got E, C, G, A, C, C, A, U, C, G. So when we're talking about a codon, this is one codon, the next three is going to be another codon, this next is going to be another codon. One nucleotide is not read twice. This is non-overlapping. And no nucleotide is left in between. This is called continuity of the reading. So these are the characteristics of genetic code which you must know. Now coming to degeneration. What is degeneration? Degeneration. Multiple codons for the same amino. Degeneracy can be best defined as multiple codons for same amino acid. I'll talk regarding this degeneration, but I will keep my discussion brief. When you talk about coding codons, you have 61 codons, which I can represent like this, like a dash, like this. Multiple codons are there. 61 codons are there, which are going to code 20 amino acids. So the ratio-wise, if you see, the number of amino acids are lesser compared to the codon. So it is very obvious that more than one codon are going to code one specific amino acid. Similar fashion, if you're talking about this particular amino acid is coded by maybe this codon, this codon, or this codon, or this codon, right? And suppose if you're talking about, this amino acid is coded by this codon alone. So this multiple codon coding the same amino acid is called degeneration. And there is something called unambiguity as well. That's another characteristic of genetic code. Unambiguity. What is unambiguity? Unambiguity. So that you must know, if one codon has coded one amino acid, as you can see in this picture, that codon is not going to code any other amino acid. Similarly, suppose this codon has coded this amino acid, you don't see any other arrow coming out of it. It's not going to code any other amino acid. This codon coding this amino acid, that's dedicated to this. Codon is dedicated to this amino acid, not going anywhere else for, coding any other amino acid. This is called ambiguity. So please try to understand the degeneration and unambiguity, which clearly you can understand using this picture. Both are different terminologies, different characteristics of genetic code. Degeneration means multiple codon coding the same amino acid. Unambiguity means one codon is coded one amino acid, it will not code any other amino acid, right? Now coming to the next characteristic, that is universality. There was a time when they used to say that genetic code is universal. I mean, they used to say that genetic one codon, with whatever the species it is, that will code the same amino acid. But now they have found certain exception to the rule of universality, which I'll be giving you in a table. So they say now that its genetic codon is nearly universal, they are not absolutely universal. So I come to that table which is talking to you. Okay, wobbling I'll be coming shortly. And this is the exception to the rule of universality. We have said you guys is stop codon, but is a stop codon for the messenger RNA, which is coming from nuclear DNA, nuclear genomic messenger RNA. UGA acts as a, but in mitochondria, it codes for tryptophan. So it's the exception to the rule of universality. Similarly, AG and AGG, everybody knows that they are meant for arginine, but they act as a stop codon in the mitochondrial messenger RNA. AUA codes for isoleucine in the nuclear genome, nuclear messenger RNA, and AUA code for methionine in the mitochondrial messenger RNA. Internal methionine is coded by AUA. So these facts you must know regarding exception to the rule of universality. Now coming back to this, degeneracy, which I was discussing some time back, multiple codons for the same amino acid. You can see the illustrative example here, which probably you don't need to remember. Proline is coded by CCU, and CCC. Alanine coded by GCU, GCC, GCA, GCG. One important point to notice is that only the third nucleotide is different, right? For those codons which are going to code the same amino acid. Similarly, for the leucine also you can see UUA, UUG, CUU, and CUG, CUC, CUA. These are going to code for leucine. So this region is multiple codons for the same amino acid, and this can be explained by a process called wobbling. So what is wobbling? Whenever there is a pairing of messenger RNA and the transfer RNA during translation process, what scientists have told that this is the codon, right, on the messenger RNA. At the third nucleotide, there is no strict base pairing which is being followed. Like generally, it must be pairing with C. That's okay, it's a normal pairing. But it's quite possible that B will pair with U as well. So this is called wobbling. There is non-Chargaff pairing that's seen at the third position of a codon on the messenger RNA. So this place there is a wobbling. And if I tell you this, the codon 3' end, right? So you see this is codon and this is 3' end. Codon 3' end, if it is 8, it will pair with either U or inosine of the transfer RNA. If it is C, it may pair with G or I. If it is D, of course, it will pair with C. It may even pair with U. If it is U at third position of the codon, 3' end, if it is UMP, it can pair with the first A, then G and I also can pair with this. So this is a table which is clearly talking about how the wobbling happens. And this wobbling actually is the explanation of degeneracy phenomena where multiple codons are going to code for same amino acid. Right. So this is all for this video. Thank you so much and stay tuned. I'll be uploading another part 2 of this where I'll be talking about further aspect of genetics, where I'll be talking about the metabolism, replication, transcription, and translation. And in part 3, I'll be talking about technologies. Thank you so much. Wish you all the best.